I'd like to thank BAUS for allowing me to talk about a subject that I actually am very passionate about. I'm a urologist, but I also run a basic science research lab. So you'll sort of see a lot of transitions between the clinical arena and what I do in the lab. This is my disclosures. Actually, nothing I'm going to present today, or actually one work that we'll present today has to do with a grant from the Greenberg Bladder Cancer Institute. So this is the disease state that we've been asked to talk about today. Obviously, bladder cancer is a disease, actually, in my opinion, has three disease states. You've got metastatic disease, you've got early stage disease, and you've got locally or locally advanced disease. This is the wealth of knowledge that's occurred in the last 3 to 5 years. Understanding the molecular genomics and intrinsic subtyping of urothelial cancer has just exploded onto the scene. But I think we have to go back and realize that bladder cancer is not just one disease. It's composed of multiple diseases with very distinct underlying biology. In 2004, Colin Dinney, who's the chairman at MD Anderson, proposed and really sort of gave a nice overview of the way to think about bladder cancer. And this has been very much validated. Maggie Knowles, who's actually from the UK, has really shown that you really have two distinct phenotypes. You've got papillary disease, the low grade, which is an FGFR3 mediated mutation-wise. You've got sort of a lesion that starts off as dysplasia and then can become an invasive lesion. You kind of think about it as CIS, or carcinoma in situ, which is very genetically unstable. You see changes in RB, RAS, P53 pathways. So this is sort of where we were for many years. And really, the thing that I've sort of taken from a lot of this work is really how we can exploit the FGFR pathway and its unique biology as it relates to treatment of bladder cancer. Because as I said, I'm a urologist, and I obviously we take pride in doing very thorough TURBTs and doing good surgery. But ultimately, if we can affect what we do in the OR by modulating biology, it's really important. So FGFR mutations in urothelial cancer are predominant in low-grade disease. This is in papillary disease. As you can see here, you see changes in mutations. Upwards of 60 to 80% of all low-grade papillary tumors have FGFR3 mutations. When you start to look at invasive cancers, this drops off to about 25, 10, 15% in invasive lesions, and this was shown in the TCGA recently. So this is clearly a unique biological pathway that should be exploited, and there are a number of clinical trials which are investigating this in early stage disease. Here's just an example of how we consider the mutational and chromosomal loss as it relates to early and then late stage disease. You can see chromosomal loss gain in higher stage cancers. You see FGFR mutations high in low grade disease, going down low, activation of P53 mutations, RB in our more advanced disease. So you can see these references. These are sort of references from 2004, 2009. But it wasn't until the last 5 years where we've really come to clearly understand the biology of urothelial cancer. And it comes from this, which is, this is an example of the bladder wall, the mucosa with the deeper layers. But we could actually substitute this with a breast bud. So this could be a breast tissue. And we know that now muscle invasive disease as well as non-muscle invasive disease can come from two different lineages. You can have development of tumors from the umbrella, which is a luminal based, and then you can have tumors that base from the basal membrane. These are more of like the stemness. You have enrichment of stemness in these basal-like tumors. So really, this is a nice framework for what I'm going to talk about in the next coming slides, which is really the molecular subtypes of bladder cancer. This is sort of the molecular genomics of urothelial cancer. In 2012, the group from Lund, really Godfrey and the group there, they were the first to publish their subtyping of urothelial cancer. And it's amazing how they were able to show things many years before others. And what this study showed was, and the reason why you may not hear about this as much is because they actually had 5 subtypes, and they were a little bit more difficult to distinguish between the groups. So it never really caught on, but it wasn't until the TCGA that was published in February 2004, which is run by the NCI, where investigators send in tumors. The tumors are then sequenced. You have whole exome sequencing, RNA sequencing. You look at epigenetics. You look at a number of different projects. And what this article focused on was the RNA sequencing of these tumors. What they found is that bladder cancer had greater than 300 mutations per tumor. You had 22 rearrangements, 204 segmental CNAs. You saw recurrent mutations in a number of genes related to cell cycle regulation, chromatin regulation, and 9 distinct genes were distinct for urothelial cancer. One of those is ERC2, CC2, which is a DNA mismatch repair mutation. And I'm not going to talk about it right now, but this is a perfect example of if a patient has an ERCC2 DNA mismatch repair mutation and they receive cisplatin-based chemotherapy, those are the patients that have been shown in now 2 groups with validation groups. So those are people that have PT0 rates in the bladder after cystectomy. So it's an example of how a cisplatin, which is known to affect DNA mismatch repair genes, is able to predict a pathological complete response. I put this here because bladder cancer is a heavily mutated gene. This is important. Very unlike prostate and kidney, it's very mutated, which means it's going to likely respond to things like immunotherapy or even chemotherapy-based treatments. This is the original TCGA that was published in Nature. There was 4 clusters. I put this little highlight here because just a couple of months ago at ASCO, or I guess a month ago at ASCO, Seth Lerner showed the 5th subtype, which is a neuroendocrine cluster. Here's an example of the way you think about clustering. You got papillary histology, that's your luminal. Remember that figure I told you about where you develop luminal papillary tumors from the umbrella cells? So this is cluster 1 is your luminal subtype. If you come here to cluster 3 and 4, this is your basal subtypes. They're enriched for genes that are expressed via the basal stemness. Here you can see that they're hot, or they're quiescent or don't have staining for markers of luminal such as FOXA1, uroplakin, or GATA3. So here you can see the way the TCGA did it. You've got enrichment for luminal markers, enrichment of basal markers. So they were divided into clusters. But it wasn't until exactly almost a month after that, the Nature article, these 2 articles came out. One was from David McConkey's group at MD Anderson. Woo-Young Choi and the group there was able to show how subtyping tumors actually predicted chemoresponsiveness. And then Billy Kim at UNC looking at intrinsic subtypes of bladder cancer and how it relates to that of breast cancer biology. This is sort of the way to think about it, and I know it looks like the matrix here, but the reality is that you can cluster using hierarchical clustering. You can break down urothelial cancer into basal, luminal, and this is the P53-like subtype that MD Anderson described. One thing to keep in mind is it doesn't mean that it has P53 mutations, but it has gene expression signatures which are consistent with P53 signaling. The most important thing I think to keep in mind here, and this is as it relates to predicting or potentially a prognostic biomarker, that tissues, that the tissue obtained at the time of radical cystectomy is if you had a basal subtype, you had a worse prognosis, overall survival, and disease-specific survival when compared to luminal and P53. I'm not going to show you today is that if you give a patient with a basal subtype cisplatin-based chemotherapy, they are actually the ones that have the most, that have a complete pathological response, or more likely to have a complete response. Here's once again how you think about basal luminal. You've got markers of basal and luminal that are enriched in these tumors. Importantly, you can look at this immunohistochemically. So you could actually come up with a nice, you could consider it almost a nomogram or a tissue biomarker that you can actually stain tumors that have luminal markers and basal markers. Now this is this slide and this figure from this paper. This is actually from Trinity from MD Anderson, the pathologist there. I think this is misleading because this is not the way urothelial cancer actually looks. You very rarely see complete immunohistochemical staining black-white. This is not binary, okay? And an example of that is actually if you look at your preclinical models. Here's an example of a preclinical model of bladder cancer where you see progression from CIS, papillary, invasive disease. When you do your RNA-seq here and then use your BAUS17 gene panel from UNC, and what you see is simply this, is that if you have a controlled tumor, or excuse me, control bladder, you see no enrichment for basal markers. But when you develop cancer, you get enrichment or activation of these basal markers. If you go by the TCGA, or at least that clear MD Anderson and UNC phenotype, you would expect to see luminal markers such as PPAR gamma, ERBB2, FOXA1. In the tumor, you would see loss of that. You don't. And this goes with the understanding that they are not, some tumors are not clearly basal or luminal. You could have urobasal-like tumors. How this predicts aggressiveness and response to therapy is really unknown at this time. So to summarize, this is our nomenclature, where you've got luminal tumors. In TCGA, they're clustering. In MD Anderson, they're luminal P53-like. In Loon, they're genomically unstable, infiltrated. And then UNC, luminal basal. This is how we are now classifying bladder cancer. If you look at why that's the case, you see in basal tumors P53 pathway genes that are activated, P53 pathway genes in the P53-like, and PPAR gamma, which is an important marker of luminal tumors. And if you knock out PPAR gamma in a mouse and you then give them or induce bladder cancer, you actually don't see papillary tumors. They actually become a basal-like. So this is clearly one of the drivers of luminal disease. Importantly, we have to look at how these immune markers are found in the subtypes. So in luminal tumors, they're called immunoquiescent, or they're not enriched. They're actually dry. They're immune desert is the way it's been described. So luminal tumors don't have activation of immune markers. But in basal tumors, you get activation of these, or enrichment of lymphocytes. Importantly, look at what's being activated in these basal. PD-1, PD-L1, CTLA-4, TIM-3. So these are actually what has been proposed potentially to be those that are more sensitive to immunotherapy. I put this in here because I think when you consider how you can do this globally, for example, in the NHS, doing complete RNA sequencing or panels is difficult, but now there's a commercially available way to do this. And this is a publication in European Urology from Peter Black and Sealer et al., where they use the Decipher platform to be able to subtype tumors. So this is something that can clearly be used in clinical practice. Moving to non-muscle invasive disease, we have actually the same way of classifying. In this publication, which was Cancer Cell last year, it was classified into class 1, 2, and 3, not luminal basal. Just to point you to the direction, here you can see class 1 are enriched for those luminal markers or histology, and class 2 has more higher grade tumors. They have more CIS. And this predicts progression-free. So if you have a class 2 tumor, at least in this cohort, this predicted progression. So when you think about BCG, it's all about terminology, because when we talk about how molecular subtyping could predict BCG, it's all about terminology. And we recently put together a consensus statement for the FDA, which looked at BCG definitions. BCG unresponsive disease is when you develop a recurrence within 6 months of last BCG exposure. That's induction BCG. So what I'm going to spend the next 2 minutes, I think I have 2 minutes, is talking about how you could potentially use this globally in your clinical practice. So we asked the question, can we predict BCG response by looking at subtype classification, by exploiting that FGFR PPAR gamma pathway, and also determining if the immune checkpoint molecules are active in non-muscle invasive disease. The study design was using a multi-center study where we collaborated with British Columbia, Peter Black, MD Anderson, Colin Denny, Ashish Kamat, and myself at Hopkins. We performed RNA sequencing now on over 200 tumors, and we developed a TMA that had patients' tumors pre and post BCG that were responders and unresponsive disease. This is, I purposely didn't put anything here to describe it because I will tell you subtyping tumors prior to BCG do not predict anything. It only tells us that you could have basal tumors that don't respond, or you could have luminal tumors that don't respond. There was no clear subtype that predicted response. But what it did tell us is that you could have switching from basal tumors became more, luminal tumors became more basal, and we're currently teasing that out. Looking at FGFR, once again, I discussed the importance of FGFR. These mutations are highly prevalent in non-muscle invasive disease. We looked at how we can use FGFR. We performed a clinical trial using davitinib. Noah Hahn was the medical oncologist that led this trial. It was a joint collaboration between urology and medical oncology. We looked at the treatment of davitinib and non-muscle invasive disease, and within 2 years, we enrolled 13 patients that had different mutation statuses. And what we found is that once again, subtype didn't predict response to FGFR-based therapy, but if you had an FGFR3 mutation, you were actually less likely to progress in this trial. Looking at PD-L1, it's highly prevalent, it is prevalent in more advanced disease, but not so much in non-muscle invasive disease. This is an adaptive immune response in which CD8 cells via TCR signaling with MHC in the bladder on bladder tumors actually are able to release interferon gamma, which actually reduces or prevents bladder cancer from progressing. When the tumor has this adaptive immune resistance, upregulates PD-1 and PD-L1, binds to their ligands, and actually suppresses CD8. This is the mechanism by which it works. So if you could develop an inhibitor here, which we're fully aware of, and I guess we'll hear a little bit about it later, we're able to reactivate that T cell helper cell. So is this relevant? Well, in metastatic disease it is, and currently we are looking at its role in non-muscle invasive bladder cancer via a number of clinical trials. They're working through the cooperative groups. Here's Pembro, which is a PD-1. PD-L1 is working through SWOG, and then there's a trial using Bivalacqua, which is PD-L1 in this disease. So we have a lot of trials being performed, but is there evidence to support it? This is the criteria to measuring PD-L1, and what I will tell you, using our TMA, unfortunately, we see very little PD-1 or PD-L1 staining in any of our tumors. I mean, we don't see it in our responders or non-responders. And as you can see, if you go by the definition proposed, PD-L1 density and those that were actually the most responsive in metastatic disease, we see very little staining in our tumors. So my perspective is that molecular characterization of muscle-invasive tumors, as well as potentially BCG, may help us guide treatment, depending on if they're infiltrated, immune infiltrated, or have a signature, for example, PPAR gamma or FGFR. And I will tell you that this is something that is gaining a lot of traction and is being investigated throughout the world. Thank you.
Thanks very much, Trinity. I can take a question or two, I think. Yeah, do we have a microphone at the front here, please?
So Trinity, the question is your signature on the primary tumor going to necessarily reflect what the metastatic lesions are doing, and how do you combat that?
Yeah, great question. So the work that we've done thus far, as well as others, have tried to correlate that, and it doesn't necessarily reflect. However, the majority of the time, so for example, if you do 10 If you have 10 tumors in the bladder that are basal, and you look at their lymph node, sequence their lymph node, 8 out of 10 times it's gonna be basal. So it's not 100% concordance, but it's pretty close. I'm waiting to see, the Loon group has put together an amazing series of this, and I'm looking forward to seeing what their results show.
I have, Simon, would you like to ask a question? Go on. Microphone here. Just while they bring the microphone forward, one question for you, Trinity. I'm a jobbing urologist in the north of England with not much access to fancy sequencing. For a T1G3 tumour, it's sometimes, I'm trying to decide, take the bladder out or give them some intracycle treatment. Are you better at doing that than I am currently? Or is that something for the future?
So I would use my clinical predictors. Is there lymphovascular invasion? Is there multifocal CIS? My restaging TUR, is there tumor that's present on my restaging? And I will tell you that as long as there is not a significant amount of CIS, and you know those bladders, we all know those bladders. And I go back on my TUR and I don't find T1 disease, I'm giving them BCG almost 100% of the time.
And is the genomics coming into that at all yet?
You are starting to see immunohistochemical characterization of those tumors.
Okay.
However, that is very much experimental and not being performed routinely. We're obviously doing sequencing to understand the biology, which I can tell you right now, I'm not learning much. I'm more confused now than ever. And that's why we expanded our cohort to 200 tumors. The discovery cohort was 30 tumors. Now we've expanded to 200, and we're hoping to be able to understand things a little bit better.
One more question, Simon, thanks.
So with seeing all this data fall out and the division into different subgroups is a little confusing in the sense that everyone uses different names.
Right.
Some people think there's 2 groups, some people think there's 5. To what degree do we just have to say, right, this is the consensus, nail it down, so that when we're devising prospective trials, we can actually at least prospectively think about a clearly agreed subset of bladder cancers?
Great question. I mean, it's a long-winded answer, but and I'm gonna try to break it down real quick. I love the fact that Roche has done their best job of subtyping their tumors from the Tezo trial and been able to show that potentially a luminal infiltrated tumor is more responsive to Tezo. The problem is they never gave anybody that data to be able to do our own. What you guys do is to actually validate that. So what has to happen is that we all have to come up with a nomenclature, which I personally believe is basal luminal. And if you're a basal, are you a Claudin low or a P53 like, or if you're a lumal, do you have P53 enrichment?
So you're getting confusing again.
No, I'm not. It's still basal luminal. The question is, how do those pathways within basal luminal help predict? I don't think you're ever going to get basal luminal. You're not. And you don't see that in breast cancer. Think about, is it HER2 positive? Not. What is the ER, what is the status, ER status? We don't know that. They use all of those to be able to predict. So I think, I don't think it's as simple as basal luminal. I think we need to add at least something. I personally believe it's the sort of the Claudin lows P53.
Great. Well, thank you very much, Trinity. That was a fascinating insight into the genomics of bladder cancer. I just want to remind the audience that if you're too shy to stick your hand up, or if the roving mic doesn't get you, you can ask questions via the app, and we've got an iPad here to pick up your questions, also by Twitter as well. So we began with genomics and urology. Obviously, urology represents a needle in the haystack of genomics. Here to contextualize the meaning of this is we have 2 speakers who are non-urologists, and we're going to start off with Dr. Sandi Deans. She is from NHS in England and is the National Laboratory and Scientific Lead of the Genomics Implementation Unit. And just before Sandi starts, or while she's preparing her slides, we're just going to do a little straw poll about the 100,000 Genomes project. So from the audience here, how many people come from a hospital that is currently collecting genomes? So probably about a third, maybe. So Joe's going to do the counting.
10.
10, great.
Give
So
or take.
So hopefully Sandi's talk will throw some new light into this fascinating project. So Sandi, thank you.
Thank you. Okay, so thank you very much for the opportunity to come and tell you, most of you, about the 100,000 Genomes Project, really the aims, why we're doing it, how we've gone about to deliver this, and also just a snapshot of a small number of results and numbers of samples going into the project. So the first steps for the 100,000 Genomes Project was way back in 2012, when the Human Genetic Strategy Group published their Building Our Inheritance paper. And that really set about the issues around genomics, the need within the NHS to be able to provide some type of genomic testing into mainstream medicine. And then later that year in December, the Prime Minister, who was David Cameron at that time, announced that the government intended to achieve a paradigm shift in the way that genomics is used across the NHS. So he committed £30 million to sequence 100,000 whole genomes by the end of 2017. Now that target has shifted slightly. We're now moving into 2018, but hopefully we are well on our way to achieving that now. And just to put it in context, the 100,000 Genomes Project is focused on both sequencing for rare diseases and also for cancer. And really the fundamental issue is to understanding the genomic basis of these conditions to help aid management of patients, and also opportunity to being included into clinical trials that may not have been possible before. So for the rare disease approach, really surgery, the incidence of rare disease is about 3.5 million. So rare disease isn't that rare, actually, and the diagnosis potentially now is very poor at present. So you have many, many families who have many, many hospital visits, clinic visits for many, many years, and still do not have a diagnosis for their affected child. And that has a knock-on impact on not just the individuals, but the full family members as well. Surgery plays a significant role both in the management and the mitigation of rare disease, and also the genomics is going to give you a greater diagnostic yield, which is what we're ultimately wanting. We want those patients to get their diagnosis, and also that has the opportunities of supporting earlier intervention, which also hopefully will save the NHS more money, which is always what we're wanting as well. On the cancer side, surgery is responsible for about 70–80% of cancer cures, and you know that better than I do. And genomics really is more about not aiding the diagnosis, but informacal surgical approach and clinical management. But before we get there for mainstream cancer medicine, we need to transform the way tumours and tissue is collected, so we're more genomic friendly, and we actually have that opportunity to get a genomic result if we need one. The project is incredibly high profile, we get a lot of government scrutiny, and this is just a snapshot of some of the committees and the meetings that are regularly taking place, and we're constantly asked to monitor our numbers and patients being recruited into the project as a benchmark of the success. So until the results are coming out and we're in steady state and we can use them for clinical management, it's more about the recruitment as how the project has been measured as a successful benchmark or not. And I think it's really important to realise that the cancer delivery plans have been part of the National Cancer Programme for NHS England, and was also part of the 5-year forward view. So it's very much on the forefront of the Department of Health's agenda. So not too much, a bit too much text on this slide, but really just a few take-home messages. So the aims and principles, as I've mentioned, is rare inherited diseases and your common cancers. The real importance is about transforming the NHS. It's not about just delivering a project, and when we reach 100,000, everything will stop. It's about drawing the patient from routine care and treating through the routine channels and actually changing the pathways so genomics becomes mainstream and is an option for every patient. Consent is a big issue. A lot of people fully endorse the fact that their whole genomes are going to be tested, and others run a mile. So it's very important that the patient is fully informed of the consent, and they realise that exactly the implications of this. The tissue and the data do not go outside the NHS-controlled safe haven, and this is very important that everybody understands that, and all the users, including the GSIPs or any of the expert scientific community, will be properly authorised and monitored, so we ensure that this is the case. And I'm sure most of you have heard of Genomics England. So Genomics England is a public limited company that was set up, owned by the government, to actually deliver the project themselves. And that really was the startup mentality and the drive to get this initiative moving. So we work very closely at NHS England Genomics Implementation Unit with Genomics England to ensure the project delivery is fit for purpose and integrated into the NHS system. The clinical and diagnostic data is collected, and it's very important that this is a unique, different type of project because we are collecting clinical data up front for that patient so we can use that in the interpretation of the results that are coming back. It will be integrated into patient records, and then in the future as longitudinal studies, and also they perhaps are re-emitted or are seen again in a different setting, clinical setting in the future, that information will be readily available. So the data and samples are collected both at the start and longitudinally through the patient's pathway. And Genomics England have a secure data centre where they store together both the whole genome sequencing results and the patient phenotypic data. So for all this to come together and actually make it a fit-for-purpose, here-to-stay project, a lot of communication and coordination and buy-in from different groups has been achieved. So we have the Department of Health to ensure it's embedded within the NHS England, Genomics England, who are the remit is to deliver the actual project and improve our genomic knowledge, as we've heard very eloquently from Trinity, that we're just starting out on that learning. NHS England itself to turn that genomic knowledge into health intervention. So what do you do with those results? How is there a need? It's not just about research and development or drug companies targeting specific patients. It's more about stratifying patients and delivering personalised medicine. But we're all learning, and we need to upskill our workforce, both from theatre staff, surgeons, pathology, oncologists, right through to laboratories and clinical genetics as well, to be able to know how to deal and how to deliver genomic medicine. And then also to take that further and involve Public Health England so we can use that knowledge in not just treatment, prevention and health protection for the whole community. So we had a quick question before the start of the presentation, was who is involved in the 100,000 Genomes Project. So we do want to get out to every geographical area. We know access to a lot of genetic tests is currently very inequitable. Depends where you are. It's very much postcode lottery. So as part of this, we want to get rid of that delivery of genomic medicine. So we have 13 NHS genomic medicine centres across the UK, covering 3 to 5 million. You can see here they're mapped out. And we have a lead organisation for each GMC. They work with their local delivery partners, so you may be part of the local hub, the GMC itself, or you may be part of a local delivery partner who have, for the cancer programme, open up different pathways depending on which tumour types, and particularly if you have individuals with a real interest in genomic medicine, then they tend to be a lot more proactive and we have more pathways open. But we are trying to get out there and encourage people to submit samples. I'm going to talk slightly about the workforce development because this is a real concern. We're all learning about genomic medicine, and it's fine to have all this data, but if we don't know what to do with it or how to act on it, or how to deliver it, then that's quite a problem. The key roles in NHS GMCs are starting from the top, is to capture the data. And this is having to be pulled out of lots of different systems. Everybody knows that there's cancer registries, different patient-specific databases, etc. So we're trying to mainstream and make it a lot more efficient to be able to automatically download that information into one single place, obviously against defined high national standards and against their data sets. The labs also have their sample collection and their processing and the logistics, and obviously, again, we have national quality standards for that. As the results are coming back out of Genomics England, we need to think about the return of the results to the participants and the clinical care, and how does that fit in with the guidelines that are currently in place? Do we need to update the guidelines? We're all learning, as I say. Transforming the NHS, improving your outcomes, and as I mentioned, reducing the variation, because there is a clear demand for that. Patient and public involvement is a very key role with the NHS GMCs, because it's not just your clinical staff that may approach the patient with information of the project, but actually the patients are coming to us and they're coming to their clinicians and saying, I want my tumour to be sequenced, can you make that happen? So there's a helpline in place, which is funded through NHS England and delivered by Genomics England to allow that to happen. Upskilling the workforce, I'm going to talk about that in the next slide. Partnerships and networks. When you speak to the GMCs, this is the overwhelming transformation and change that people have seen. They say, we're actually talking to people now. We're talking to our oncologists. We're talking to our surgeons. We're talking to everybody within the pathway in pathology. And we're understanding their part in the pathway and how important it is, and that you can't do it on your own. You need everyone every step of the way. And that has been the most overwhelming response to GMC, their view of the project. So there needs to be a step change in analysis of genomic information, the validating and reporting, because we do have molecular pathology testing and molecular inherited genetic testing, but not to this degree, not to cover single nucleotide variants, copy number variants, mutational signatures, structural variants, all in the one test. So there's a huge amount of information, and we need to deal with what's important, what do we report, how do we report it. And the reporting is not just down to the scientists. It's very clearly been demonstrated that you need multiple clinical specialities to be able to interpret these results, because you always have to take it back to how the patient is at that point of time when you have the result. Whereabout in the clinical pathway are they, and is it appropriate to act on it? So it really is to have multidisciplinary team discussions to take the results forward. So this is just an example of the pathway we have in place. So we have our recruitment and consent. The samples then go to the centralised GMC hubs, where they bring together the IT and the clinical data that's required. They process the samples for DNA extraction, and we've done a huge amount of work and improving and enhancing DNA extraction methodologies and measurement of samples and QC steps. So that has improved not just 100,000 genome samples, but also your mainstream genomic testing that's happening here and now. So that has an impact already on patient care in the NHS. The phenotypic data, as I say, has to move with Genomics England, so they can use that in their interpretation of the whole genome sequencing results. And then those results come back into the NHS remit for the validation and the reporting out to the referring clinician. So Health Education England Genomics Education Programme have set up a lot of some training and online modules, and really that is to support the NHS England staff, because as I say, this is all very new for everyone, and really with the aim to have more of a long-term effect for capacity and capability. So when genomic medicine becomes more mainstream, and it is here to stay, that we have that capacity and our workforce is educated appropriately, and that's going to lead to the lasting legacy of the genomic medicine. Some of the resources that are available is the consent conversation. Obviously, that is very important that the consent is appropriate. And there's online courses. We've got 3 units, genomics and healthcare, the project protocol, so to give you an idea of what's happening, and also the patient-clinician conversation. And here you see down on the right-hand side, we've eligibility wheels, can't say that word, for the rare diseases to find out whether or not your patient would be accepted into the project. go. So the feedback from, just as an example, the consent conversation, we have had 2,500 people registered since February 2015, and you can see that comes from all types of background of staff members. And after completing the module, about 95% said that they were ready to go out and actually felt confident to have that conversation with their patients. So what about the surgical role? In rare disease management, you already play a key role in the management of your patients. And for the urologists, obviously polycystic kidneys is one of the main rare diseases that we recruit to. And we really want to, and in the cancer side, we know that 1 in 2 of the population are expected to develop cancer at some point in their life. And cancer really is a lesion of the genome. So that is why the genomic testing is so important to be delivered for your patient. So part of it should be key part of the activities across all surgical specialities. And that's why we've tried to be inclusive with as many cancer types as possible within the eligibility criteria. So why whole genome sequencing? Well, that sequencing of the whole genome is 3D mapping of your cancers. So you're actually finding out what's happening not just on your string of DNA, your actual sequences with your single variants, but you're actually looking at structural variants, your copy number variants, and looking for particular mutational signatures to actually give you such a better understanding of your tumour that you can then guide your clinical management. We have, sorry, I'm got muddled up now. So we need to get the pathology right. If you don't have a genomic-friendly tumour sample, you're never going to get a reportable whole genome sequencing result. Now, we have a lot of whole genome sequencing data coming back now from the pilot phase of the cancer programme, whereby we were only collecting fresh formalin-fixed, paraffin-embedded tissue. And we know that there's a lot of chaotic DNA sequencing is being produced, and there's very limited amount of reportable results coming out. It really is quite overwhelming, the difference that fresh frozen samples make to getting a reportable result. So the DNA extracted, we know that the paraffin-embedded formalin is not a good way of treating your DNA. It gets degraded, it's heated up, it really adds mutations into your DNA. So how, when you sequence, do you know whether it's been introduced due to the handling, the tissue handling, or whether it's actually there in the first place? So there's been a huge move to fresh frozen pathways across all of the tumour types. And particularly, we've just identified 3 of the GMCs as accelerator cancer sites, and one of the remits of those GMCs is no FFPE tissue to be submitted into the project at all. Now, embedding this pathway does require a huge amount of change, both in sample handling, right from the minute the tissue is excised through to the time it goes to pathology. And there's a huge drive now with the Royal College of Pathologists, Genomics England and NHS England to move formalin out of theatres. So not move formalin out of pathology or hospitals, but specifically theatres. And there's many areas and hospitals who are now doing this. This is an example of the pathway that currently exists. So you go from surgery into pathology for cut-up. It goes on to formalin fixation, paraffin embedding, then your cutting and your IHC, your H&E analysis for diagnosis, DNA extraction, and on to any molecular testing that's available. And really we want to bypass that and follow the red arrow, going straight from cut-up to fresh tissue and DNA extraction if a diagnosis is confirmed. So it's just giving that patient an opportunity to have genomic-friendly tissue at the start, that if it's required and that diagnosis is confirmed, then they can go on to whole genome sequencing. There was a lot of concern about the HTA and how the handling and storage of tissue would work with the current status. So we've worked very closely with the organisations listed above here to issue a consensus statement, and hopefully most of you will have heard and read this statement. And the main difference is that tumour tissue samples, so that's fresh, frozen and FFPE, and the blood samples for germline DNA are to be considered as diagnostic samples. So you don't need a specific HTA licence to store your fresh tissue for the defined number of days. That it's classed as diagnostic, so that is absolutely fine. And this has made a huge difference to a lot of the clinics and trusts to be able to implement their pathway when this was going to be a really big issue and they weren't perhaps a large teaching hospital that had an HTA licence. So just to snapshot some numbers before we finish. So for the recruitment of families into the rare disease programme for the renal and urinary tract, we've now got 1,297 families, that's from last week, being recruited, and you can see the breakdown here. I don't need to go through them, but it's nice to see it's quite a high number of families. So that's not individuals, the majority of them will be trios, so that's number of families. For the cancer, this is the weekend in the 21st of April. You can see the renal and bladder I've just highlighted. So these are individuals, so we've got 421 renal cancer individuals recruited and 76 bladder. But as I say, this was at the end of April, so the numbers will have gone up because the pathways are opening every week as we speak. Some of the snapshot of some of the results, and this is just hot off the press from last week. So here is the pink is the variants in actionable genes, and you can see bladder here is pretty much 100% of the results that are coming out of the project to date, whereas renal is about 50-50 with or without actionable genes. So just to give you an idea, the results, the variants that are detected are classed into 3 domains. So domain 1 are your actionable variants in the 72 actionable cancer genes that is currently available, and that is tiered into relevant to your tumour type. Domain 2 are then the 590 cancer-associated genes that it's up to you whether or not you validate and whether it's relevant. And then domain 3 is everything else, and that's where a lot of the researchers and the GSIPs want to triage that data and find out what's interesting. And then, very nice following on from what Trinity was showing us earlier, this is some of the mutational signatures that we're starting to see. Again, it's still small numbers just now, but we can pick up quite a lot of the common mutational signatures, particularly in breast and colorectal and lung. And you can see we popped in prostate and bladder at the bottom as well for you. So you do know the results are coming through. So really just very final slide to say we are on the up. Today we're treating our patients with a one-size-fits-all, but really we want to go forward to whole genome sequencing being mainstreamed, and it will be commissioned from next year, 2018, from the autumn, for particular referral reasons. And that is the way we're moving to make this a transformational project, so it's here to stay and it does become mainstream genetics. So thank you very much.
Sandi, that was a great overview. Just in the interest of time, and just because Eamonn's talk I think is going to be complementary to yours, I think we'll take questions together after Eamon's talk, if that was okay, but thank you for that. So our next speaker is Eamonn Maher, who's a professor of medical genetics and genomic medicine at Cambridge, having moved there from Birmingham in 2013. Now, of particular relevance to this audience is Professor Maher's critical involvement in the elucidation of the genomics of kidney cancer. He was responsible for identifying the VHL gene and several other kidney cancer genes. So it's a genuine honour to have him speak today to us with the tantalisingly titled Dream Sequences in Genomic Reality.
Thank you. So the title I better explain. For a clinical geneticist to be able to access high throughput next generation sequencing is a dream, but I was asked to also focus on some of the challenges and limitations of genetics in practice. So I'm going to talk about current application for genomics for both research and for clinical service, and just point out some of the issues that we have in terms of our everyday use. So genomics can be used for a variety of applications. As a clinical geneticist, we see people who have or thought to maybe have inherited disorders. So we're mainly focusing on sequencing of blood, but my own particular interest, as we've heard, is in inherited cancers. So we may also be involved with analysing their tumours. And the reason for wanting to identify individuals who may have inherited forms of cancer is even though they're uncommon, it's important for the management not only of them, but their relatives who may be at high risk of developing tumours. And being able to recognise them and test them and then intervene with surveillance programs significantly reduces morbidity and mortality. So we can recognise individuals presenting with cancer who may have an inherited form of cancer, perhaps by the presence of syndromic features such as cutaneous leiomyomas in patients with hereditary leiomyomatosis, renal cell carcinoma syndrome. They often will have a family history of cancer. On average, they'll have an earlier age at diagnosis, and they may have multiple primary tumours. But none of these particular indicators in itself is enough. And indeed, patients with multiple primary tumours may have them for environmental reasons because of the radiation or the chemotherapy that they have had. So this can be quite a challenging group. So recently we've been looking at this group and saying what's the potential for doing very comprehensive genomic analysis on them. So we initiated this multiple primary tumour study through the NIHR Rare Diseases Bioresource Bridge study. So we aim to recruit individuals who had 2 or more primary tumours aged below 60 or 3 aged below 70 that had negative genetic testing in NHS diagnostic testing with or without a family history. We've recruited over 700 patients, and to date we have whole genome sequencing data back for just over 400. And so what we've started to ask is, what is the yield, diagnostic yield, of using whole genome sequencing of the blood in these individuals? So what's the extra positive results that we can pick up as a result of using whole genome sequencing above and beyond the routine NHS diagnostic sequencing they'd have? And as you can see, these are not in any way predominantly urological patients. Breast cancer is the most common tumour, but there's a huge variety of different combinations. And so one of the plans was to look across here and see whether we could identify new genetic causes of cancer. But initially, as I've said, we have just looked at those known inherited cancer genes and see whether we can pick up unequivocal mutations in those patients. And to cut to the chase, we have about 15% of patients in whom we've identified a germline mutation. But now to move on to the limitation and challenges here. The gene names are color-coded by background. Where there's a dark background, that means that the cancers that were associated with this were the typical cancers we would predict. So, for example, colorectal cancer with these mismatch repair gene mutations. But there are a number of genes where there's a light blue background, and that means in those cases, the tumor that was found in that patient, or tumors, were not what was expected to be found from the known tumor phenotype associated with those genes. So either we are uncovering new tumor genes associations, or potentially some of these findings are just coincidental. They just happen to have a genetic susceptibility, and it's unrelated to the cancer they develop. So this is very recent data. We're still working through it, but I can take you back to an example we published a few years ago where we identified a new tumor gene association and illustrate some of the challenges that we face with this. So there's quite an overlap between genes that predispose to kidney cancer and those that predispose to pheochromocytoma and paraganglioma, particularly for succinate dehydrogenase mutations. So pheochromocytoma is one of our interests. We were referred a patient who was a 6-year-old boy with pheochromocytoma, had the routine genetic testing done in the NHS laboratory, and not identified any mutations. So we proceeded to a whole exome sequencing. And the variants in the exome that was of most interest to us was a missense substitution. So a single amino acid was substituted in the fumarate hydratase gene. And we were interested in this because we knew this was an inherited cancer gene. And it was the function of this gene is very closely related to succinate dehydrogenase, which is a major cause, when defective, of predisposition to pheochromocytoma. Fumarate hydratase is known to be associated with hereditary leiomyomatous renal cell carcinoma syndrome, okay, which is characterized by cutaneous leiomyomas, very aggressive papillary type 2 kidney cancers, and in women, uterine fibroids. There was no reported history of this in the patient. He was very young, however, but even in his family, there was no history of this. So we went on to look at this variant in a bit more detail. And we also screened some additional patients and found another one who also had a rare variant in the fumarate hydratase gene. And when we find variants in genes, we try to look and see whether they were already reported, whether they've occurred in normal individuals. For both of these variants, they hadn't been reported. But they could just be coincidence or they could be pathogenic. In this case, we were suspicious they were pathogenic because if you look in other organisms, even down to zebrafish, Danielle down here, the exact amino acid was present in the fumarate hydratase, that gene. So working with one of our colleagues, Christian Frezer, we were able to show that if you take mouse cells that were deficient in fumarate hydratase and put a normal fumarate hydratase gene back, intracellular levels of fumarate were depressed. But when we put in the 2 mutations that we'd identified, we had no depression, suggesting they were loss-of-function mutations. So this would be consistent with a new gene tumor association. And as we were doing this, there was a publication from a French group who'd identified 5 patients who'd also have pheochromocytoma and paraganglioma. So now we've moved that into clinical practice. We routinely screen our patients with pheochromocytoma and paraganglioma for fumarate hydratase mutations. They only care about 1% of patients, but clearly, clinically, it's important because we believe they may also be at risk for renal cell carcinoma. One of the puzzles we're left with is if we look in red where the location of the mutations that are associated with pheochromocytoma are, and compared to the location of the mutations, those that are associated with hereditary leiomyomatosis, there's no rhyme or reason to this. They're spread throughout the gene. So, made progress, but there's still some unanswered questions. So I've talked about whole genome sequencing and whole exome sequencing. When we do whole exome sequencing, we only sequence about 3% of the genome, so, but that 3% contains the regions where most of the disease mutations will be found. So at the moment, it's still more cost efficient to do whole exome sequencing, although it's not as sensitive as doing whole genome sequencing. So for NHS diagnostic laboratories, doing whole exome sequencing has been challenging because of costs. But a few years ago, we decided that we wanted to move towards doing whole exome sequencing, and a stepping off point might be to do targeted clinical exome sequencing. So what I mean by targeted clinical exome sequencing is that we don't sequence the coding regions of the 20,000 genes that we would do with with exome sequencing, but we just do the 5,000 genes that have been known to be associated with human disease. And this was using an Illumina TrueSight 1 assay. So we introduced this after a validation period in February 2014. So this was for blood testing for patients that were referred to our genetic service with a wide variety of possible inherited disorders, including inherited cancer syndromes. And what we found was that we were able to use this in a very cost-effective way. So when we did a health economic analysis on it, we found that the cost of sequencing using this, what we call Gemini, Genomic Medicine Initiative, was £650, whereas the average cost for the test that we've been doing before, and that mainly often involved sending away to other laboratories, was just under £900. So there was a major move across to this. And although we sequence almost 5,000 genes, the way that we use it is just to analyze those genes that we think are clinically relevant. So we actually analyze virtual gene panels, and that reduces the frequency with which we identify variants that we're uncertain whether they're relevant or not. The diagnostic rate is over 30%, And when this was introduced, it meant more or less that any genetic test that we wanted to do, we could apply with this. Over the years, some of the more recently described genes haven't been represented on this panel, but there's a new assay just being released that now includes those and now includes about 7,000 genes. So as clinical geneticists, we've gone from thinking, how can we find a pathogenic variant? Which is the gene that's most likely to be the cause, and we should sequence that first, which is what we would do when we were doing single gene variants, to actually finding lots of variants, and then asking ourselves the question, have we got the right variant? Is this variant disease causing in this patient? And is it an incidental finding, etc.? So just to discuss some of the limitations of this. The whole experience has been very positive, but there are limitations. And one of the major limitations is variants of uncertain significance. So these are variants that are rare. We can't say they're benign, but we don't know that they're pathogenic. And this grey area can be a real challenge to deal with, both for clinicians and for patients. And if you get the classification wrong and say it's benign when it's really pathogenic or vice versa, that can lead to major problems. So there are a variety of ways that we can deal with this. As more sequencing was done, we get more knowledge and it becomes easier to classify variants as benign or significant. We can have international collaboration that will exchange data so that we can more reliably classify them. And there's projects such as the ClinVar projects to do that. That we can use tumor sequencing if we're dealing with inherited cancer syndromes. And for ourselves, because of our very keen interest in succinate dehydrogenase mutations causing pheochromocytoma and renal cell carcinoma, we've gone a little bit further and developed some tools that we think are helpful to do it. So one is to use immunohistochemistry, so just standard immunohistochemistry. And this was first described by a Dutch group about 6 years ago, and what it was described was as if there's a germline SDH subunit mutation, almost invariably, or almost always, there is a loss of SDHB protein expression in the tumor. And so we can do immunostaining on the tumor, and that will give us a good indication whether or not that's likely to be a pathogenic variant or not. So we now have this as part of our standard clinical armamentarium. We've also been investigating the use of metabolomics, both in vitro and in vivo. And this is still at a translational research thing. But when you inactivate the succinate dehydrogenase gene, what you do is get a buildup within the tumor of succinate. And if we have fresh frozen tumor, we can use HR mass analysis to pick up a succinate peak. And we've done 47 SDH-related tumor types now and shown a very clear difference between those tumors that have a succinate dehydrogenase mutation and those that don't, including some tumors where we initially didn't find an SDH mutation, but found elevated succinate and went back and showed that there was a problem there. And we've done a small series of about 12 patients where we've been able to use NMR spectroscopy to pick up succinate within the tumors in their body. And so that also looks quite promising. And once we've done this for a particular variant, then we've shown it's pathogenic, then any other patient that is found to have that variant, we can reliably categorize that mutation. Oh, sorry. So this was almost the final point to make, which was that we had a patient here who was 18 years old, presented with a pheochromocytoma, with an extraadrenal pheochromocytoma, a paraganglioma, with excess noradrenaline in plasma catecholamines. And they were sent off for genetic testing, and the result came back negative. Now, we know about 40% of patients with a pheochromocytoma have an inherited cause, and that's even higher in young onset cases. So we were highly suspicious that there was an underlying cause to be found. So either we weren't testing the right gene, there may be new genes to be found, or the test that we used didn't detect all mutations. And we'd use the clinical exome testing here, but this doesn't pick up deletions and duplications. So we asked them to do a specific test looking for this, and we found a mutation, a deletion in the SDHB gene. And really the point of reinforcing this is that clinical picture should always guide your genetic investigations. And if there's a discrepancy between them, then you should question the lab very carefully about it. So high-throughput genomics is fantastic, but there are a number of challenges that remain to be solved. So we must be able to have our sequencing strategies that will detect all mutations. Whole genome sequencing will be a step towards that. We need to make the turnaround time for our testing much quicker. It's typically 2 to 3 months in routine now. And if we're going to make an impact on acute medical care, it has to be much faster. We need to be more reliable in our interpretation pipelines and develop more complementary methods to interpret variants of uncertain significance, such as immunohistochemistry. And we also need to think about how non-clinical geneticists can be supported in interpretation. And this has been alluded to already. So what we do in our own practice is we have multidisciplinary meetings with the laboratory where they bring the DNA variants to the meeting, and we interpret them in the context of the clinical picture. And that may result in further testing being requested from the laboratory. It certainly filters out some of the variants that are found, and it then proceeds to molecular genetic reports. And clearly we can invite other specialists to come in and join these for particular groups of patients. So just to finish off, I'd like to thank the people who've done the work: James Whitworth, Ruth Casey, Madhu Bassetti, Ferdy Gala, etc. Thank you.
Great, thank you very much for that, Eamonn. So, Sandi, I don't know if you want to just step back up, and we've got a few minutes for some questions. There's actually a question that's come through on the app. This question is probably directed to both of you, in fact. An important point raised early in the talks was not of only personalised treatments, but also of early interventions. Given the likelihood of features found on sequencing being clinically relevant to multiple disciplines, whose responsibility is it to coordinate the patient's care across the disciplines, particularly if these other disciplines are not already involved in the patient's care? Nice, simple one to start off with.
So I think that will probably depend on the particular condition. So something like von Hippel-Lindau disease, which is a special interest of mine, because it's multiple specialties involved, it's actually often fallen to the clinical geneticist to coordinate the ophthalmologist, the urological involvement, the neurosurgeon, surgical involvement, etc. Where there's a single specialty involved, for example, inherited bowel cancers, such as familial polyposis, it's often the gastroenterologist that will do the follow-up because they will be undertaking the endoscopic investigations. So, and if we were talking about inherited eye disease, then care of the patient is usually undertaken by the ophthalmologist, but testing of the family would often be undertaken by the clinical geneticist. So I think there won't be one size fit all, but it will vary from condition to condition.
I would just echo that, and it's a clear example of how we need to have multidisciplinary team working and interact in that communication that I was talking about. That we need, it's now patient pathway rather than your particular patient. Yeah.
Can I take that a little bit further, perhaps be a little bit devil's advocate? As a regular victim of the MDT for a couple of hours a week, I really relish the concept of bringing even more data into that MDT discussion. It's incredibly time-consuming. Where are we going to end up with this? Are we all going to be in so many meetings about the individual patient that we have no time to treat the rest?
No, totally agree, and that's something that's been raised again and again. So what a lot of centres are now doing are setting up tumour boards, whereby the data, so the data comes back to your accredited laboratory that triage the data and work out, as Eamonn says, which ones are pathogenic, which ones are not, whether the gene is relevant or not. So there's a couple of levels. One, whether the gene's relevant or not, and then whether or not those variants that have been detected are disease-causing. And then when that information's been tidied up and neatly packaged, then goes to either the tumour board or a genomic MDT, and the patient information is then discussed, and either it goes on to be validated for variants, or it then goes on to the tumour-specific MDT, where that information is then portrayed. Because I know many of the MDTs, they go through patients, you know, 1 a minute or so, and you just do not have time to do
and
that.
we also play MDT tennis in the north, which
lung,
bladder.
Yeah.
I think we have a question right from the back there, is that right? Yeah, okay. Microphone, thanks. Could you just introduce yourself and then ask the question?
Yeah, it's Tom Walton from Nottingham. It's just a question for Sandi, please. Ourselves and other groups have published on the difficulty of obtaining genomic samples from radical prostatectomy specimens. I noticed that you had 400 cases from prostate. I take it that most of those will be channel TURP specimens. I mean, do you have a view on how we should be, is the best practice of getting genomic samples from radical prostatectomy specimens? Because clearly that's a different patient population.
So I'm not a pathologist or a surgeon at all, so I'm out of my comfort zone here, but I do know that prostate is one of the very difficult tumour types to sample, and we have quite a skewed collection of prostate samples. It's not every GMC submitting, it's only a small subset, and to reflect that, and we do get a lot of people asking, well, what's the best way to approach it? How should we be doing that? So in conjunction with the RC Path College, we're holding a tissue handling workshop in the middle of July to actually specifically talk about tumour-specific issues and share some learning, because there are people out there that are doing it, and they're doing it well, and there's others that really would need to share that learning. So you can get in touch with us and we can pass on that information if you wish to be part of it.
David Manson-Barr, Registrar in Stevenage. To your point about prostate, I spent 2 years of my life using formalin-fixed tissue on prostate biopsies with quite good effect. We got quite a lot of genetic data out of it. With the Genomics England moving away from formalin-fixed tissue and moving on to fresh tissue, how do you envisage this project moving into smaller hospitals where the infrastructure isn't available to collect fresh tissue because the logistics is frankly a nightmare getting tissue from theatres to be processed in a timely matter? And going on to your point about prostate, you can only use radical samples because you've got enough tissue. If you want to use biopsies, prostate biopsies are a wonderful thing to be testing, but handling those in a fresh environment is very, very difficult. So is Genomics England moving to help smaller hospitals develop these facilities?
Yeah, absolutely. So we have been doing quite a lot of experimental work, and I don't know if you know Professor Louise Jones from BART, she's been leading on this. So she's been looking at lots of different ways that we can help protect that tissue in transportation and logistics and just fitting in with normal pathways. So for example, she got a lot of data on using cold ischemic time, so putting samples in the fridge, even just popping on the ice and just protecting that during transportation to a central laboratory for extraction, or even within a big hospital from surgeries, putting a fridge in surgery and they just pop all the tissue samples in there, and then the porter comes and does 2 collections a day, for example, and take them down to pathology. Vacuum packing is another way that she's demonstrated that you can protect that tissue sample. Then you're not talking about big pots being stored in lots of fridges, etc. So there is quite a lot of data out there that demonstrates it's and it doesn't affect the morphology as well, because the last thing we want to do is in any way upset the normal pathway and that diagnosis for your patient. That is ultimately your main aim.
Thank you very much. I think excellent talks from both of you. Thank you. I think this leads on rather nicely to our next section, which is a debate, really. There's clearly a tension here a little bit between the information we might be able to glean from genomics, but fitting that into our already quite highly stressed clinical pathways. So our next section is about is a debate about, is the NHS really ready for genomics? And our first speaker is Simon Crabb, who is an oncologist from Southampton, who's leading for bladder cancer and the 100,000 Genome Project. He's going to be arguing for the case.
Are we going to do a vote before?
Do you want to do a vote?
Yes.
Okay, Ravi wants to do a vote. Everybody wake up. Ravi's going to do a vote.
So just because obviously we need to detect the swing by our speakers today. So we have a hand show now for who thinks the NHS is ready for genomic testing, having heard the speakers before. No one. Oh.
Two.
What?
Two. Sandy, I'm not sure we're allowed to accept your vote. only
Three people.
three so far, okay. Sandy, take note. And Simon?
Easy to win.
You're starting at the mountains in the Tour de France. Off you go.
Thank you very much. So this is just like a general election. You haven't got to win, you've just got to get a bit better than you were before.
But you're Jeremy Corbyn, so
Exactly
you're doing all right.
Thank you very much. Thanks for asking me to speak. Here's some disclosures, and here is the answer. So hopefully you can make that out at the back. My multidisciplinary team is very friendly. We all get on, and some of my best friends are urologists. And what's clear to me is that urologists are people who will take on new ideas and will integrate them into the care of their patients if it makes sense and if it helps those patients. Which rather begs the question of why are we having this debate? And I think there are three main reasons, and I'm going to cover those, and then I'm going to go on to talk show some examples of how we're actually already doing this. I am going to use genomics in a very broad sense for this talk, and almost exclusively I'm going to talk about somatic work rather than germline work. The first reason we're having this discussion is simply it's got cheap, comparatively speaking, to how it was a decade ago. So we can now sensibly think about how we might use tests of this nature in our clinical practice and think about costs that are not completely dissimilar to cross-sectional imaging costs. The second reason we're thinking about this is simply that, and partly because of the cost and partly because of technical advance, we're starting to see information falling out on every tumour site you can think of that is revolutionising the way we're thinking about the biology of these diseases. I was going to talk about bladder cancer falling out into different diseases, but Trinity has done this for me and described beautifully how this is a multitude of diseases, and if we're going to advance treatment of bladder cancer, we're going to have to think about it as a group of diseases that behave differently, that respond to systemic therapies differently, and also local treatments to the bladder itself. The other interesting point, I think, and this is just a snapshot of some data that happens to all come from TCGA, but it's actually not just common diseases. So it's not just thinking about how we can exclude the pussycats from the tigers in prostate cancer, but we're starting to see genomic data falling out for adrenal cancer that's bringing forward ideas that you could test in prospective clinical trials. We're also, in addition to common kidney cancer, seeing it for rare versions of kidney cancer. And this data is allowing us to think about rare cancers in a way that simply wasn't viable before. And the third reason we're having this debate, I think, is actually because we are already doing it. We've heard quite a lot about the 100,000 Genomes Project. We put the first bladder cancer patient into the Genomes Project last year. They then had to take a picture of themselves and put it online, because, of course, if it's not on Twitter, it didn't really happen. But what's interesting about this is it's not, when I think about the Genomes Project, to be perfectly honest with you, largely I think this is a really cool way of getting some research done without having to pay for the sequencing. But actually, this is a genetic test being done, genomic test being done on the NHS. This is an NHS test, and it says so on the website. Findings will be fed back to the NHS. A clinician then gives the findings to the participant. They will discuss what the results mean. So for those of you working in centres that are sending off samples, you will, and it's already happening, have results come back to your centre, and your centre has agreed to work out how to relay those results back to the patient. The way we've done it in Southampton, and I think this is probably the way it's going to have to be done in every centre, is we've set up a cancer genomics MDT, which now meets on a weekly basis, that takes these results, looks at them firstly from a QC perspective, which is important, but also then thinks about what it is they then tell the urologist about the bladder cancer patient that was sampled a couple of months ago, and what it is that's relevant that can be handed back to the urology MDT, because you're absolutely right, we are not going to sit in urology MDT and understand what that data means. These are some of the results that have been coming out, simplified to a very large degree. Here's FGFR3 abnormalities in bladder cancer. And this is happening now in centres that are doing this work. If you think about genomics testing more broadly, there are plenty of examples where this has been going on for quite some time. Here are some examples from oncology. In non-small cell lung cancer, if you have a fusion abnormality of the ALK gene, it makes your cancer sensitive to a drug called crizotinib, which targets ALK fusion abnormalities in non-small cell lung cancer. ALK fusions only occur in 5% of non-small cell lung cancer. So if you give this drug to everyone with lung cancer, it will be useless, it won't work. But if you do it in a therapeutic trial that first selected patients on this basis, then you can see benefit over standard treatments. Here's melanoma, a disease which not too long ago was viewed by most oncologists with a high degree of nihilism. We are now able to select out those patients that have a specific point mutation of the RAF oncogene, and in those patients use an established therapeutic which improves survival by targeting that signaling pathway. And then finally, breast cancer. And breast cancer is interesting because they've been doing it for a while. In that disease, 15 to 20% of patients have abnormalities as a result of genomic alterations in the way that HER2, a cell surface receptor, behaves. And as a result of that, they're susceptible to drugs such as trastuzumab and TDM1. So the use of those drugs is only viable if you select that subgroup of patients out. And although we do it actually by immunohistochemistry and FISH rather than a more complex test, this is still a genomic test. It's understanding the underlying biology of the disease and targeting the therapy to the appropriate patient. This has been going on routinely in all of our hospitals for over a decade. So the NHS is clearly ready for genomics. It's just a question of the specifics of the test. If you don't do this, some drugs and other therapies will fail. Here's an example that we salvaged. The epidermal growth factor receptor is abnormal in a proportion of non-small cell lung cancers. Gefitinib is one of an example of a number of drugs that target EGFR and block signaling. When the original phase 3 trials were done, at the turn of the millennium, they were initially done in unselected populations, and the trials were completely negative. Subsequently, it's been possible to show and bring into routine practice that if you determine which patients have a mutation that activates EGFR, then you can select out those patients that will respond to drugs like gefitinib, where outcomes are better than prior treatments. Which I think is important in the way we're going to move forward in prostate cancer. Here are the new drugs for prostate cancer, which this audience will be very well aware of. And they've altered significantly the way that we treat this disease, and patients are living longer as a result. These are major advances, but each of these was a trial in an unselected population of about 1,000 patients. And I'm going to put it to you that if you spend that much money and make that much effort, you can't really claim to have made a profound difference if all you have done is add 2 to 5 months to the median survival. You're going to have to do a bit more than that if you want to claim what you're doing is transformative. And so we need to do more trials like this. This is TOPARP. This is a study in prostate cancer and castrate-resistant disease, where everyone was given the PARP inhibitor olaparib in an attempt to target abnormalities in DNA repair mechanisms that are found in some prostate cancers. And in this study, the drug given to an unselected population was modestly effective, these patients over on the left. But what was possible in this trial, because they went and did fresh tumour biopsies on every patient, showing that you can do that in the NHS, at least in a clinical trial. And they were able to define a biomarker composite signature for those cancers that had an abnormality in DNA repair that to a very large degree selected out those patients that benefited from the drug. What they then did was critically important. We didn't move on and do another phase 3 trial in 1,000 patients without selection, sit tight and hope for the best in a few years' time. Instead, we've done a second trial where patients are selected for the access to the drug on the basis of having the biomarker. In other words, you do it the other way around. You do the biomarker testing first and test those that are positive, with the idea that at least at the research level at the moment, we're selecting out those patients that shouldn't have access to this drug because it's unlikely to help and they're simply going to be exposed to toxicity. This is the way to do drug development, and the UK has led this particular example. I'm an oncologist, so I tend to talk a lot about drug trials, but I think it can also modify the way that we do surgery. Here's an example from prostate cancer. This particular test, which was led from the UK, looks at a list of genes relevant to cell cycle and has shown in the expression profile of this group of genes that you can modify on top of what Gleason score does to determine a radically different likelihood of subsequent relapse and progression. There are other examples in prostate cancer. We could get bogged down in detailed discussion about whether this is the right test or not, and that is still falling out. But it's very clear that if we're going to move on, selecting those people that don't need to be cured and ideally would never have known about their prostate cancer from those that need a curative intervention, then we're going to have to understand the biology of the disease, and we're not going to do that by thinking just about Gleason scores and staging. Here's another example. This happens to be colorectal cancer, where outcomes are significantly different depending on the presence of circulating tumor DNA. In other words, doing this by a blood test. And this makes the point that we're going to have to think a little bit about where we get our samples from. Some of this is going to be fresh tumor biopsies. Some of this will still be FFPE, but some of it's going to have to come from the blood, at least as a way of getting access to samples sequentially as often as you might want. And also to think about tumor heterogeneity, which is an important point, I mean, perhaps particularly in prostate cancer. Here's an example in prostate cancer where we're doing similar things. The androgen receptor in castrate-resistant disease becomes abnormal for a variety of mechanisms, but one key example is a splice variant which leaves the resultant abnormal androgen receptor constitutively active, and you can detect this in blood. And at least at an experimental level, this almost completely matches on to those patients that benefit either from enzalutamide or abiraterone. So having used these drugs routinely on the NHS and spent thousands per month on every single patient, this offers the opportunity to select out those patients that will or won't benefit. And I would suggest that the NHS needs exactly this sort of approach to try and advance the way we're currently doing hormonal therapy for this disease. And then it feeds back into our clinical trials. Here's an example of a biomarker-directed clinical trial in bladder cancer that was launched a couple of months ago and will be open in most of the academic centres in the UK fairly soon. And we're going to select out different approaches to treatment based on underlying biomarker evaluation, which will largely be done through a targeted exome sequencing approach. So to summarize, genomic tests are just tests. They're not special, they're not different, they're just tests. And we should use them when they aid our clinical decision making, when they allow us to make smarter decisions for our patients, when they allow us to avoid pointless treatment of patients that won't benefit, and vice versa. We should, of course, be careful about the functioning, the characteristics of the test itself, sensitivity, specificity, and so on. But we're used to that with tests that we do routinely. This is no different. There are multiple examples where this is already being used and is a routine part of current practice within the NHS. And therefore, I think it's very clear that we should vote for the proposition. Thank you very much, and I'll leave you with this visual metaphor. Thank you very much.
Thank you very much, Simon. Some persuasive arguments. Vincent, you're up next. So our next speaker is Vincent Ganapagarsam, who is a urologist from Cambridge, those of you who don't know him. And Vincent is going to argue to the contrary, that perhaps the NHS is not yet ready for genomics. Vincent, over to you.
Thank you very much, Joe. So I guess if Simon is Jeremy Corbyn, I must be Theresa May. I don't think I've ever had that comparison made before.
Can we have the slides on the main screen, please?
Oh, still got the
Sorry,
apples.
Vincent,
Yeah,
okay.
sure. So when I was given this topic, I made the case that I'm not trying to defend genomic medicine in general, because then Eamonn is going to be looking for me when we get back to Cambridge and telling the Regis that I should be sacked. It's about urological cancers. And I guess I'm trying to follow on from the talks we've had today, which are fabulous and really incredibly illuminating as where we're heading. Is a really hard thing to do, but I was heartened by the 3 votes from this audience about what I was going to say. So thanks, guys. But the rest, for the 3 of you, I hope it's going to be 0 by the end I'm in this. But anyway, when Simon was talking, I was very conscious that he had to be a bit of a used car salesman. He had to bring out the best of the best, because actually that's what you need to drive this, the best of the best. I don't have to do that. I have to tell you what's underneath the bonnet. So, is it ready? Not yet. Because we can see Mount Precision Medicine up there. It looks fabulous. It's great. We're going to get up there. But we're here at the moment, urologist. That's the oncologist hurtling to his doom there. But hopefully the road will be constructed before he gets there. Now, before I go any further, just to be absolutely clear, I have been involved in biomarker, molecular, genetic studies for many, many years. I don't come from this just from a clinical perspective. And this is just an illustration of the outputs from Cambridge in the last few years, and we are key contributors to the ICGC. So really, it's not a Luddite's view that I'm bringing here today. I'm actually here to ask the very specific questions about genomics for all, because I'm not talking about genomics for the few or genomics for the little bit of people who inherit cancers, because the debate here is the NHS ready for genomics? So let's consider then, what will we assay? Who will we assay? When will we assay? How will we use it? Can we afford it? And this is the important question: will it improve the outcomes? So genomic biomarkers, I like to think of it as the evolution of Star Trek, really. So when I started doing stuff, it was like McCoy here. Individual biomarkers discovered by direct effect. It was slow, but it was elegant. You looked at one marker at a time, you switched it around, it did something. So it was like trying to find a single path through a jungle. And then everyone realized that actually that one path wasn't doing the trick. You had to combine these. So this was a bit of an illumination. And now we have these highways here, which are much straighter. But then now we don't even actually need to know what's in that path. We actually just need to take a whole bunch of genes, mix it up together, come up and see which is the one that actually shows the most promise, and that one is the one we're going to use to profile. So it's a bit like spaghetti junction. You eventually get there, but you don't quite know how. And genomics implies DNA. That's how it comes from. It's a genome. But actually, there's lots of other types of biomarkers. We've heard about a lot of them today. You can do the genotype, the SNPs, the inheritance or the acquired. You can look at tissue-based immunohistochemistry. You can look at circulating tumor DNA, including CTCs. You can even look at urine markers and exosomes. But we've not heard about a lot of this today, and the 100,000 Genome Project is focusing, as I understand it, on the tissue and the blood, not on the rest of the stuff. So where will all the rest fit in? Will we have enough money left to do all that interesting stuff? And let's look at one example of this. This is from Trinity's talk, partly from Trinity's talk. This is a study which looked, what they did was they took 5 different classifiers and tested it in a single set. Now, why bring this out? Is to show you that these all were predictive of an outcome, which was looking at survival. But they all had slightly different compositions of what was what. And we heard a bit earlier on about basal versus luminal. Then there's all these other different types in here as well. So which one is correct? Are they all correct, or are they all wrong? It gets even more complicated in prostate cancer. So this is a slide which one of our clinical lecturers produced for a review. I think he was trying to show that we are heading towards the new horizons, but actually, I found this fascinating. All these circles represent lots and lots of genetic tests which have all been shown to predict biochemical relapse. And you can see immediately the overlap between any of these gene sets is actually pretty small. So they're all right, but they're also all wrong. So you could choose any one of these and apply them, and you would be sort of right and sort of wrong. And the problem with this is not cheap. It hasn't stopped companies like Prolaris and Oncotype selling this directly to patients and physicians and telling them that we can change your outlook by selling you this test. Look at the cost here. Can we afford this? I don't think so. Renal cancer, same problem. Look at this gene list taken from this paper. I don't know much about renal cancer, but this looks like prostate cancer to me. You've got all these genes which are all been shown to show outcome. You can do it in the tumor, you can do it in the host, you can do it in the circulating biomarkers. Which would you like? Which will you use, which will you not use? So, do we really know what we're going to assay? What about who will we assay? Now, this country believes in health for all. It believes in equality no matter who you are. Therefore, every patient who has a urological cancer should be able to avail of a genomic test. Would you agree? If you're going to offer it? Otherwise, why do it? So let's look at the scale of this. This is prostate cancer 2012-2014. You can't see this. It's 39,560 patients here, just for prostate cancer. If you combine prostate, kidney, and bladder cancer, which is there, in 2007, they made up about 28% of all of all male cancers. So we're not even female here. By 2030, it's going to be 34%. So 1 in 3 of all male cancers of a urological type, let's just say we only ever did genomics for urological cancers, would really be eligible for or should have a genomic test if you're really going to bring it in. How are we going to do this? And really, will we do everybody? Here's this chap, he's 90 years old, he's in a wheelchair. He looks pretty cheerful, but I bet you don't want to be doing a cystectomy on him. And then you have your spry 50-year-old lady. Now, she may have the muscle-invasive tumor, or she may have the non-invasive tumor. Genomics, NHS being ready for genomics means you need to be able to offer both these guys the same thing. How are we going to do this? Who will you select? You're starting to see now that actually, if you bring this in without thinking about it, you're going to end up in a situation where you cannot deny people things. And we know this because MRI for prostate cancer has shown what happens when you bring in a test and then you say, through the media and other ways, that this is the best thing since sliced bread. You can't then rationalize it in your centers by saying, well, I can only afford to do it for 70-year-olds, or you look like you might need a radical treatment, so therefore you're the one who's going to get MRI. You cannot do this. You either do it or you don't do it, or else you're going to be hauled up in front of a court somewhere for prejudice. So when will you do it? Well, this is a very elegant assay. I'm not going to ask you to read it, but in effect it says you get your tumor there, you do your genetic signature, tumor biopsy, sequence it, tumor-specific genetics, all fantastic stuff. You get your clinical assay, personalized assay, breakpoint identification, PCR, all fantastic technology. And then you use it to monitor clinical outcomes just using blood tests. So it's a very nice model, actually. It's what we would like to do in the future. And that's what the 100,000 Genome Project, as it stands, is all about. Now, to be clear, I see this as a snapshot of what's going to come. But we cannot focus on that. We've got to focus on what's going to come afterwards. And you can see the investment here. We've heard about it. I think it's a fabulous idea. I think it's slightly misdirected because it doesn't consider the consequence, but that's because I'm debating this point. But it is a snapshot. It is a snapshot of a point in time when you have done that assay. Because we know that as the tumor evolves, it acquires mutations. And those mutations become different after you treat a patient. So when will you do this genomic assay? At the point of diagnosis, we've heard about that. But what happens when the tumor mutates to adapt to the treatment you've given it? You're going to do it again then? And then you're going to do it again after that? In prostate cancer, we see that happening. The primary tumor and the metastatic tumor have a different profile altogether. And that profile can be split into 2 different types. That changes after you give the treatment. In renal cancer, a nice study here, which looked at the levels of a few different genes pre and post-treatment in ctDNAs. And you can see they all go up. So your profile changes, your tumor changes, your patient changes. You cannot just do a snapshot at one point and say that's it. So when will you do this assay? We've heard about some of this very nice work looking at neoadjuvant therapy and non-responders and responders. And we see that if you classify by basal luminal and 2 different types here, you benefit some patients. You can identify those who respond and those who don't. If you look at the data, this red line represents the basal type. This blue line represents the luminal and T type. And they get worse, this gets better. So it makes sense to say, if you know who they are, that you actually don't give it to one and you give more to the other. But this study showed no value in 60% of the cohort. So even with the stratification, you still can't improve it for more than 40%. And in fact, in one of your slides, Simon, I noticed that you were talking about the TOPAP trial. But if you looked at the survival benefit stratified by the markers you said, the survival was only 6 months better. That wasn't a huge lot greater than the 3 to 5 months you mentioned. So even with biomarker, even with stratification, you're going to help the few and not by a huge amount, at the moment anyway. And let's talk about what's involved with personalized breast cancer. Joe, you were mentioning this a few times as though it was a personal threat on your horizon. Well, let me take you through the personalized breast cancer program, which has been established in Cambridge as one of the country's first integrated molecular MDTs. And this is slides courtesy of Carlos Caldas and Jean Abraham. It's a feasibility study at the moment, and I'll just quickly take you this. You get patients with early or metastatic breast cancer, you get a study consent, you get blood and tissue taken, whole genome RNA sequencing, so this is more comprehensive than the 100,000 genome. You get your maps of genetic profile, and then you go to this thing called the Oncogenetics Review Board, which I guess, Simon, is what you're talking about in Southampton. This is just for breast. And then it goes to validation, you decide what drugs to do, and then it goes for clinical trials. So all of this has been set up just for one organ. And this is what that team consists of. You got a pathologist in there, an oncologist, clinical geneticist, the cancer molecular diagnostics laboratory sitting in there, the patients, the clinical informaticians, the scientists, and the trainees. There's no surgeon there, have you noticed? This is what you need to put together an oncogenetics review board to look at the data you're getting. You can't just simply get a report about an actionable mutation. I'll come to that in a minute. If you really want to run this, you need to know what your data is saying. Because one day, that 100,000 genome has to be devolved to the centers to be able to do it themselves. Is the urology MDT really ready for this? So yes, the test of genotics is going down. What about all this costs? Who's going to pay for all of that? This is Dorchester, by the way. Anyone from Dorchester here? Apologies for using a picture. It's the only one I could find of a urology MDT. You look hopelessly optimistic, actually. If you look at you see the notes in there. That's exactly how things look like.
Friends, they're all awake.
They're all awake for the picture. So the cost of genomics is definitely falling. But once you find the mutations, you cannot ignore them. You cannot say to somebody, oh, by the way, you've got bladder cancer, you just had surgery, and you've got this mutation. There's a drug we could use, but I'm not sure we can afford it. Really? You're not going to you need to do something about it. Now, the cost of cancer drugs is very high. And the more precision drugs you made it, the higher the price, because the target population is smaller. This is a graph from America showing the cost of cancer drug development rising year on year. And I don't know whether you can read this, but these are some of these targeted drugs here. 70,000 a course, 120,000 a course, 40,000 pounds a course. Are we going to open the Pandora's box to start something we can't actually complete? Again, the analogy of MRI scans. The country urologists, we're struggling to provide what is no doubt a good thing, but we just cannot afford it. And we are already in the deep doodads. So how are we going to put this on top of that? So do we know where the need is for genomic tests? Now, if you take something from the standard pathway, you start with pre-intervention, efficacy, effectiveness, you explore the context, you adopt it, you implement, you sustain and evolve. I somehow cannot help but thinking that the 100,000 genomes and genomics in general has been sort of dropped in there, and we're having to adopt and prepare for it without any of these steps happening already. Because you cannot just have a concept. It's individualized. Every disease type has to have its input into how you do this, because one size does not fit all. So we are in a current situation where the urologist is really running to keep up with the Genomic Express. Of course, the oncologists are sitting there smiling broadly because they're already on the train. So I don't present these things to tell you that it's all doom and gloom. It's actually an opportunity, actually, to shape the agenda. Did you know that they're doing genomics for dogs? So all those vets up and down the country are also going to have the same problem. So what can we do? Because actually this is where the critical need is, and I really think Genomics England needs to wake up to this. They have not engaged some of the most important people. I'm sure it applies to all cancers. We are, as urologists, and oncologists actually, can define the clinical need. What is the population that really needs to have the tests? What's the optimal cohort? What is the best test and sample to use? Is that urine? Is that blood? Is that tissue? You know, we've heard about the problems with radical prostatectomy. We have 15 years experience in Cambridge of analyzing and looking at prostatectomy samples. We have an optimized harvesting protocol. Not once have we been approached to share that with anybody. We have actually submitted samples for quality analysis and never received any news back. What's the clinical infrastructure to apply the profiles we generate? You cannot have a single molecular board for all cancer types in a hospital. You have to devolve this because otherwise it never becomes embedded. And you need to optimize and standardize the clinical predictive tools because your genomic test has to sit within the clinical context. And we're already not using a lot of the prognostic models we have. And the most important thing is to provide space in our working lives to explore these new ideas and practices, not just left to the ivory towers chaps who sit there and pontificate about how fantastic this test and that test is. Look what I can do with this stuff, and you need to do it now. It's not for people like that. It's for the everyday urologist or oncologist sitting in a busy clinic. For every patient, you should be able to use this. And therefore you make it accessible from the start, whether in Dorchester or in London. And let's not forget that clinical
Mrs.
pathological
May, you've got 1
parameters
minute.
remain the strongest predictive markers yet, without doubt. And an example of the fact that we haven't reached the optimization, this study we published, taking standard NICE criteria, and we developed a new prognostic index with a much higher ability to predict survival. We're not even getting better at the clinical stuff before dropping in all the genetic stuff. So, I conclude. The road is not yet built, but maybe it will be, and one day we'll be the guys riding there, but at least we will not fall off the trip. Thank you very much.
Vincent, sterling effort on the front foot. Okay, we haven't got much time, so I think we better have another vote, hadn't we, Ravi? Yep. Show of hands then. Is the NHS ready for genomic testing in urology?
Change the question. Still 3.
So Sandy still believes in it. Well done, Sandy, for sticking to your guns. And there were 2 more hands at the back there, yeah? Oh, Simon's up there. 1, 2, 3. So, Jeremy, I think you're more or less where you started. But, you know, it was the lion's den, wasn't it? So, Vincent, Simon, Trinity, Eamonn, and Sandi, thank you very much for an excellent and illuminating afternoon. I hope it's been interesting to you as well. And we must make way now for our young researchers who are probably the future for you for 2025, I guess.